All right, guys. So good morning, everyone. Good morning, people that are watching also from the YouTube platform. Is a great honor, joy, everything else to introduce the first speaker of our course. A great, good friend and a person that I admire so much, and he's been with us since the beginning of this course, about 25 years or so, Shay.
Yeah, I know.
Yeah, around that. So normally, and you will see from the next speakers, we do kind of a background information, so on and so forth. But I must say that the first time that I introduced Seamus about 25 years ago, it took, sorry about the airplane. So it took me around 5 minutes to introduce all Seamus' honor, achievements, and everything. And he has it recorded somewhere in his room or office, and it's quite embarrassing, actually. You know? So I'll just say that if you want to check, you just go Google Scholar and you just be amazing. So suffice today to say that if I had somebody to recommend as a mentor, it would be Seamus. Okay, so please, and you can see the face of this guy with the glasses, Ronaldo. He's going to be with Seamus for his PhD. How happy he is. So Shay, thank you so much, really, to be with us always, from the beginning.
Thanks, Gustavo. And good morning, everybody. And yeah, it was great to have Gustavo and Karina in Ireland just a couple of weeks ago. We had good fun and lots of good chats as usual. So it's a pity I couldn't be in Brazil today, but I guess we're here. I'm there in spirit. I've been there many times and I really always enjoy visiting. So okay, without further ado, I'm going to share my slides if I can remember how to do that. It's been a while, as I say, since I've given a Zoom talk. Thankfully, we all got too used to this, obviously during the pandemic, but luckily real life has started. So let's see. I don't actually see. Sorry, it's
You see your share screen, or you can.
Well, this is a new. So I have to give permission to Zoom to do that.
Oh, Márcio, a gente precisa dar permissão.
Ele já está com permissão.
Yeah, you already have the permission, Shay.
It says, it's from. It might have to leave the meeting. It's to reopen. It's saying unless I quit and reopen. Oh. I just updated Zoom. It maybe got to do with permissions. I'll just try again. Let me just try again here. Okay, I can see. All right, I can see it now. So.
Yep.
You see it, yeah?
Yeah, but not in the present. Yes, now.
Yes.
Now. Perfect.
All right, great. Okay. Yeah, so I'm going to give a bit of an overview of regulated cell death. I'm not going to talk about all forms of programmed cell death, mainly the kind of major one that we know about apoptosis. And I'll touch upon necrosis, unregulated necrosis, and maybe later on also I'll talk a little bit about regulating necrosis. But what I'm going to just give you, first of all, is a bit of history on, how we used to think of cell death. And because the field really, in terms of the study of regulated cell death or programmed cell death, it only really took off in the 1990s. Before that, there were very few groups that were actually working on this field. And most people didn't really study cell death because they didn't think that cell death could be controlled in any way. And now we know that obviously that's not the case. When cell death occurs, it's almost always controlled. And I'm going to get at really why that is. What's the major reason for regulating cell death? Why is it important to regulate cell death? And I'm going to give you the answer to this question up front. And the answer is the cell death machinery seems to have evolved in order to minimize the damaging consequences of eliminating cells. There's lots of situations that I'm going to tell you where we need to kill cells for various reasons. And when you do that in a deliberate fashion, when you deliberately get rid of cells, it's very important that cells just don't burst open, because when that happens, you trigger inflammation. That in a nutshell is why you need regulated cell death. You do it to regulate inflammation. So when you want cells to be removed silently, they're eliminated via apoptosis, the major form of regulated cell death. And when you want cells to activate the immune system, there are types of regulated cell death that are programmed necrosis, where you deliberately rupture cells to release molecules that are very good at activating the immune system. Now, I'll get into that as we go on. So, okay, if I, let's see now, is this working? All right, so I work on Trinity College in Dublin. It's a very nice campus here in the center of Dublin. And this is the Liffey running through Dublin, and we're right on the coast. And it's a pretty old campus. It was established in 1592 on this very, campus. Not all the buildings are original, but, many of them are. So it's a pretty nice place to work. And if you haven't visited Dublin, I would encourage you to do so. So that's another view of the main quad within the campus. Okay, so as we know, the cell is the basic unit of life, and there are many, many cells within the human body, and of course within all multicellular organisms. And multicellularity has evolved so that cells can cooperate to achieve more than they can individually. And I know that's all very obvious, but there are certain conditions that really must be met before cells can basically get together and cooperate for the purpose of achieving something that individually they cannot do. And these are things that I think are pretty obvious, but we don't often think about them. And certainly, I'm not aware of a textbook that really discuss these things in a lot of detail. So there are some fundamental rules in a multicellular society, which the body is, of course. And of course, rule number one is, you do your job. So if you're going to cooperate with other cells, typically what happens is there's a division of labor. So not all cells do the same thing in a multicellular organism. Particular cells specialize to do individual things. I mean, so that's essentially the fundamental bargain. If we get together to cooperate, then I'll do certain things and you do certain things. So that means I'm going to give up doing things that I was doing as a single cell, and now you're going to do certain things that I'm not going to do, and I'm going to do certain things that you're going to do. So it's really important that you do your job. It's also important in a multicellular organism that if you differentiate into become a cardiomyocyte or a T cell or a neuron, you pretty much do what you're supposed to, and you stay where you're supposed to stay. You don't get to wander around the rest of the body. Another fundamental rule, of course, of multicellularity is you don't eat your neighbors. So if cells are going to get together, they have to trust one another. And so, that is a very basic rule, that you don't eat your neighbor. You don't build, another property. So, as homeowners, we know that, you need permission if you want to build an extension to your property, or if you want to build another home on your property. And the same thing happens within the body. Cells don't get to divide unless they have permission from other cells, and that permission is given in the form of growth factors. Another rule is you don't get to harbor viruses or bacteria inside. So, because otherwise that could threaten the multicellular cooperative. And finally, and I'm just going to, encapsulate this, is don't act weird. You don't, for example, make proteins or peptides that you shouldn't make. And that they're pretty fundamental rules. I think they hold up well if you really scrutinize them. And the reason I mention all these is because if cells don't obey those rules, then typically they die. They're killed by programmed cell death. And I'm going to get into, some of the ways that that's achieved. But fundamentally, if cells break those rules, if they start moving where they shouldn't move, if they start dividing when they shouldn't divide, if they get infected, if they display foreign peptides, i.e. act weird, if they don't do their job, then typically they're killed by programmed cell death. So that's a number of the situations where it's very useful to have a means of executing cells on demand because of one or more of those reasons that are mentioned there. So just again, summarizing it, in a cellular society, if you don't perform your job, if you're injured or infected, abnormal or dysfunctional or breaking the rules anyway, then typically death is the penalty, programmed cell death. And that's one of the reasons we have a programmed cell death machinery, is to get rid of cells in those various circumstances. And as I mentioned at the outset, to get rid of them in a way that minimizes the negative consequences for the rest of the cells in the multicellular organism, that minimizes inflammation. So programmed cell death enables cells to be eliminated with surgical precision. And I'll return to this later, but I'm just showing an example here of apoptotic cells, the red cells here that have these highly condensed nuclear fragments within them. This is very characteristic of apoptosis. So the cell here at the bottom left, in green here, the mitochondria are labeled green and the nucleus is labeled blue. These are viable, healthy cells. Well, cells undergo apoptosis, so the red color is propydium iodide that's entering the cell because it's dying. And then the blue, you can see here that the nucleus has become hypercondensed and broken up into these, much smaller fragments, which again is very typical of apoptosis. So apoptotic cells have a very characteristic morphology, and those cells are undergoing a highly programmed type of cell death. So because new cells are born on an ongoing basis in many tissues, they get injured, they get infected, they become dysfunctional, become aged, and then they die in regulated manner. There is a constant throughput of cells, being born, going through this aging process, and as I say, getting damaged or injured or dysfunctional, and then they die by programmed cell death. And there's a huge amount of that happening through our lives in our bodies. So we have about 10 trillion cells in our tissues, and it's been estimated that, they're turned over about 1,000-fold in our lifetime. So we make a lot of cells, basically about 10 to the 16 cells. So there's a huge amount of cell division going on and a huge amount of cell death. And that is very finely balanced so that, cells are born when they should be born and they die when they should die. That isn't left to chance. And it seems amazing now in retrospect to consider that we thought cell death wasn't regulated. This amount of cell death, if it wasn't regulated, would have very serious negative consequences for the body. Again, to reiterate, as I mentioned earlier, cells die really for, breaking obvious rules or due to injury or infection. And there's also certain special circumstances where they die because they only need transiently during the building of other structures. And we're going to get to that, in a moment in the context of development. Another observation we can make is that tissues that suffer the most damage exhibit the highest rates of cell death. Of course, you'd expect that. And those tissues also exhibit the highest rates of cell division. So these things are very interrelated. We know intuitively that the skin, the lungs, the gastrointestinal tract we know these tissues are, prone to the highest rates of damage because they're facing the world. They're more exposed to constant day-to-day damage, exposure to environment, exposure to infectious agents. These are our major barrier surfaces. And so there's an ongoing predictable rate of damage in those tissues. And as a result, cells in those tissues are very short-lived. And so they undergo turnover at a very high rate, approximately 20 to 30 days, is basically the lifetime of a cell in the skin and gastrointestinal tract. And when you think about it, in the adult, if we had no cell death in tissues, we wouldn't need cell division. We're very familiar and comfortable with the idea that there's a lot of cell division going on in tissues, but you only need cell division in the adult because you've got cell death. So cell death comes first, and then cells divide to replace the cells that have died. So there's as I say, this fine balance between the rates of cell division and the rates of cell death. And, that balance needs to be struck throughout life, or disease results. If you have too little cell death and too much cell division, essentially you get cancer, or other diseases that result from accumulation of extra cells, such as inflammatory diseases, as we'll again mention later on, or I'll show an example in a moment. So I'm just going to go through then some specific scenarios. We see cell death taking place massively during development, during the sculpting of limbs and the building of tissues. A very common theme is that you overproduce cells and then you kill off cells that you don't need within that population to leave you with the mature cell population. And there's some very striking examples of that during the development of the immune system, where, for example, you massively overproduce developing T cells called thymocytes, or developing B cells, and then you subject those cells to a test, and then only the small fraction of cells that pass the test, so about 5% of the T cells that you make during development, actually get to enter the peripheral immune system. So 95% of the cells that are produced on the way to developing a T cell population don't actually get to pass the test, and they're killed off, and they're killed by programmed cell death. It's very similar to B cells as well, and also developing neurons in the brain. Typically, you massively overproduce neurons, and then you let those neurons wire up and find each other and become innervated. And then many of the neurons fail to make proper connections, and they die by programmed cell death. So it's a repeated theme in the immune system, during development, that during the development or creation of complex structures, you overproduce cells, and then you select the cells you want from within that population, and you kill off the rest of the cells. So we see programmed cell death being used as a sculpting tool repeatedly during development. And then in the adult, as I've already mentioned, the homeostatic control of cell numbers in tissues. Clearly, there's a massive amount of cell division that happens during development just to build the organism. But then when the organism is developed and mature, you still need a fairly high rate of ongoing programmed cell death to keep the tissues healthy. So cells are turned over at a predictable rate related to the amount of damage or infection that tissue faces. And of course, some tissues, as I mentioned a few minutes ago, face a lot more damage than others. And that leads me to damage and infection. That's a situation in the adult where you see a lot of programmed cell death. Typically, the easiest thing to do when a cell gets infected is to kill that infected cell, shut down the infection by killing the cell. And as we're going to see, of course, we have specialized cells called cytotoxic T cells or natural killer cells that do that very effectively. And also cells have ways of sensing damage, particularly to DNA. And those damage sensing pathways typically result in programmed cell death. It's better that you simply eliminate a cell that has suffered damage, particularly to DNA, rather than trying to repair that damage. Because if the repair is faulty, then you may get a cancer as a result. So in most cases, when cells are damaged or infected, they're simply killed by programmed cell death. So just an example there's developmental sculpting of the limbs. So the limbs initially develop as these primitive limb buds that get larger and larger. And then you have a limb paddle. And then finally, the interdigital cells in the paddle start to die off to create the, the fingers and toes that you get in the as the embryo proceeds to maturity. You build tubes, blood vessels, and other tubes within the body, again by a process of overproducing cells. So initially the tube is solid, and then the cells in the middle of the tube, they die in a process known as cavitation, and then the tube becomes hollowed out as a result of that programmed cell death process in the middle of the tube. I mentioned earlier developing T cells and B cells. They undergo this process of selection, in this case, on the basis of their T cell receptors or B cell receptors being able to appropriately interact with the antigen presenting structures called MHC molecules. And clearly, if a T cell fails to recognize MHC, altogether, then it will die by neglect. And again, that's a programmed cell death. If it too aggressively binds to MHC or MHC push cell peptide, then again, it will undergo negative selection and die by apoptosis. And that's the fate of the majority of developing T cells. And then really it's T cells that recognize MHC, but in a not too high affinity manner, they're positively selected, and they're allowed to migrate to the periphery and become mature T cells. And cells that have slightly higher binding to self-antigen then can become regulatory T cells. But of course, this process of selection, would be impossible to implement any other way because this is based on random reconfiguration or recombination of T cell receptors. And again, something very similar happens with B cell receptors that undergo this process of random configuration by shuffling different T or B cell receptor segments together to generate an almost limitless conformation of those receptors. You end up with lots of T or B cells that have reshuffled those receptors inappropriately and they need to be eliminated. So typically, I mentioned repeatedly now that infected cells are typically eliminated by programmed cell death. And that's, typically done by cytotoxic T cells or natural killer cells. They use slightly different ways to kill target cells. But in essence, what happens is target cells are infected. It's presenting a foreign antigen on its cell surface in MHC. That's recognized by the T cell receptor of the CTL. And then that triggers the CTL to either degranulate its cytotoxic granules onto the surface of the target, and then that delivers certain enzymes that we'll mention later on that activates the programmed cell death machinery in the target cell. Alternatively, there are membrane ligands called Fas ligand or trail ligand, or indeed TNF ligand, that can be present on the surface also of the CTL or NK cell, and that can engage cell surface receptors on the target called Fas or trail or TNF receptor. And that can also activate the programmed cell death machinery. So the two distinct ways that our body has evolved to kill cells that are infected, or indeed that are just presenting non-self atypical peptides, or indeed that have lost their MHC molecules. And there's an example of what happens when genes that are, in this case, one particular gene that is expressed that's involved in shaping T and B cell populations. And that gene is a molecule I mentioned a moment ago that's encodes the Fas receptor. So this is what happens when the Fas receptor is mutated on T and B cells. These are normal lymph nodes here on the top left. And then after about 16 weeks in a Fas receptor null mouse, this is the size of the lymph nodes. So you get massive accumulation of extra T cells. And on the right-hand side here is what happens in a Fas knockout mouse to the spleen. And there's a normal spleen here on the top. So there's massive accumulation of excess T and B cells as a result of the failure just to eliminate that T cell populations that undergo expansion every time they encounter antigen. So there's a very heavy price to be paid if you don't regulate cell death. Then cells will accumulate that are defective or autoreactive or indeed infected. And that's why it's really important we have a mechanism to regulate cell death. So I told you some of the why do you need programmed cell death, and now I'm going to kind of move into how. And by the way, feel free if anybody has a question. I'm very happy if you want to just ask a question as we go along, or if you want to elaborate on something I've said, or hear some more about it. So do feel free to just, interrupt.
Anybody has questions at this point? Otherwise, you make notes and we discuss everything at the end of the Seamus talk, okay?
Okay, so I'm going to go fairly quickly through the how. So we've said lots of situations, I've discussed lots of situations where it's important to be able to kill cells or for cells to kill themselves in various circumstances. So let's take a look at, how this occurs. And let's dig into the detail of why I think cell death is regulated in the way it is regulated, because there are different ways you could achieve the same result. So first of all, let me deal with necrosis. So as I say, until about the 1990s, when people talked about cell death, they refer to it as necrosis. And pathologists, who do these things clinically, when they look at cell death in patients, they typically talked about necrotic lesions. All cell death was considered to be necrosis, and necrosis is kind of a car crash where cells pretty much explode. We now know that exploding of cells can take place in a non-controlled manner. That's just conventional necrosis. And that happens in lots of situations where, for example, if cells are exposed to high concentrations of drugs or toxins or extremes of temperature, or indeed, just physical injuries, if you fall and you scrape your, hand along the ground, then you're going to kill a lot of cells just through that, physical interaction of the tissue with, the hard surface that you fall on. So necrosis is just a rupturing of cells very suddenly, and it happens, as I say, under those extreme conditions. And that's very similar to a car crash. It's not planned, it's very sudden, it's very chaotic, and in many respects, no two programmed cell deaths are the same. They happen, in a very circumstance-specific manner. So there's kind of nothing to learn from studying necrosis. In the same way that there's very little to learn from studying a particular individual car crash. It doesn't happen in any kind of controlled manner. Now, we used to think all cell deaths take place like this. And of course, we know that that is not the case when cell death is programmed and regulated. There's an actual dedicated machinery that's switched on that coordinates the process of cell death to achieve a regulated cell death. So the types of cell death that we fundamentally recognize now are the kind of unconventional necrosis, which is akin to cell murder. Cells have events thrust upon it. The cell doesn't participate in its own death at all. And as I say, is seen in response to high concentrations of chemicals or toxins or temperature. And then we have regulated cell death, which is like a cell suicide. Cells actually participate, they receive signals, and then they turn on the programmed cell death machinery, which we're going to see. And then there's a type of cellular execution where cytotoxic T cells or natural killer cells can deliver enzymes into a target cell to switch on that cell's programmed cell death machinery. And that's also apoptosis. So we've got necrosis, unregulated cell death. We've got apoptosis, which is regulated cell death. And we've also got CTL and NK killing, which is also regulated cell death. Let me deal with necrosis first of all. I've alluded to this earlier. It doesn't really matter if you just want to get rid of cells because they're infected, or because you need to eliminate them because they're autoreactive, or you need to eliminate them to build a blood vessel. Doesn't matter how they die. Well, it does matter how they die, because if cells die by necrosis, that's very damaging to their neighbors. So if you're going to regulate the cell death, it's better that you don't simply switch on some mechanism that bursts the cell open. Now, we have programmed necrosis, which I won't go into a lot today, but there's a programmed necrosis called pyroptosis. There's something called necroptosis and ferroptosis also, which are considered to be forms of programmed necrosis. And this happens in situations where it's actually beneficial to release cell contents to get a very powerful immune response. So, but that's an exception. So normally when cell death is regulated, it happens by apoptosis, which doesn't allow release of all of the intracellular contents. So the bottom line is, if you want to kill a cell in a regulated manner, it's better that it doesn't happen by necrosis, because dead is not dead, right? In that, there's a type of cell death that's damaging, and there's a type of cell death that's much less damaging for the body. So necrosis is very damaging because, of course, when you rupture cells, you release lots of enzymes that are listed there: lipases, proteases, DNases, RNases, carbohydrases, and so on. And you also release molecules that I'm going to discuss briefly now in a moment, called DAMPs. And DAMPs are molecules that are very good at activating the immune system. I'm going to tell you a little bit about the history of the discovery of DAMPs. But the bottom line is, in addition to nasty enzymes that kill other cells, when you burst the cell open by necrosis, you release DAMPs, and DAMPs activate the immune system. That's really what you want to avoid when you eliminate cells in a programmed manner. You want to try and minimize collateral damage and death to neighboring cells, because remember, as I told you earlier, cell death's happening in all our tissues all the time, just homeostatically. It's a way just to keep the tissue healthy. Cells are turning over in a very predictable manner. It's not some kind of an extreme event. If you have an extreme event, you have a big infection, or you have a burn injury. Okay, in that situation, you want to mobilize the immune system. But if cell death is taking place in a very kind of, stochastic, constitutive manner at a low level, then you don't want that to continually activate inflammation in your tissues. So in most cases, the regulated cell death that we get is apoptosis. And apoptosis really is designed to minimize release of intracellular contents, minimize the release of DAMPs, and not activate the immune system. So the concepts of DAMPs was introduced by Polly Matzinger about 25 years ago now. And basically what she realized is that the immune system responds to more than foreign antigen as such. In fact, foreignness isn't the major determinant of how the immune system is activated. We used to think that the immune system discriminates between self and non-self. And to some degree, that's true, but it's not completely true, in the sense that we know we can synthesize non-self peptides, inject them into a mouse or ourselves, and get absolutely no immune response. The immune system does not recognize a peptide as being non-self unless another condition is met. And that other condition, Polly Matzinger kind of posited, was danger. So non-self triggers very effective immunity once the immune system has basically detected something that's that is indicative of danger. And what is danger? Well, Matzinger suggested that danger is either infection or a lot of tissue damage. That's what danger is. And so you see non-self as long as that's qualified by either a clear indication of infection, and that effectively is a PAMP, which was put forward by January. And PAMPs are, conserved components of infectious agents that can activate PAMP receptors, such as Toll receptors. Well, Matzinger's contribution was realizing that cell damage releases analogous molecules that she called danger signals, now called DAMPs, that also can interact with some receptors and macrophages, and are good drivers of immunity. So I'll explain that a little bit better in a moment. But essentially the question is, how does the immune system know when to respond? Does it respond to non-self? The answer is No, non-self is not enough. As I mentioned, you can take a peptide. It's totally foreign. Immune system will not respond to it. If you put that peptide in along with an adjuvant, and what adjuvants effectively do is what we're talking about, they cause tissue damage and they release these endogenous molecules called DAMs. And so Polymasker suggests that non-self in the context of danger is very good at driving immunity. And in fact, just danger itself, so you can have necrosis in a completely sterile situation, and that's very good also at driving inflammation. So essentially, apoptosis is an antidote to that. In apoptosis, everything that happens is going against the release of DAMs, because in apoptosis, you do not want to activate the immune system, as I mentioned. So again, just to underscore that concept, in infection, infectious agents are associated with pathogen-associated molecular patterns, such as LPS and CpG and peptidoglycans and so on. And those PAMPs qualify any antigen seen in the context of that infection as dangerous. And typically you get then a strong inflammatory response that's geared towards eliminating infection. And similarly, when you get an injury, when molecules are released from tissues, intracellular molecules, which are called DAMs, again, they're powerful activators of the immune system and drive inflammation either in an infectious setting to eliminate infection, or in a non-infectious setting to drive wound healing. So basically, apoptosis is all about avoiding the release of DAMs from cells. And in programmed cell death, most of the events that happen basically minimize the release of DAMs, because DAMs are very powerful activators of macrophages and really are very apical initiators of the immune response. So again, the outcomes are healthy cells are ignored by the immune system. Necrotic cells indicate danger and start to ramp up the beginnings of a powerful immune response. And apoptotic cells, as we get to, they minimize the release of DAMs and facilitate the recognition and removal of the apoptotic cell from the tissue before it can leak open and release DAMs. Just want to touch on DAMs for a moment. What do we think the major DAMs are? This has been debated for quite some time. We think they're members of the extended IL-1 family, and don't have time to go into why we believe these are the major DAMs, but I've written a couple of reviews on that in recent years. And basically, the IL-1 family members all fill the kind of the expectation or criteria you'd expect to find in a DAM. And they signal in a way that's very, very similar to PAMs. So these are the members of the extended IL-1 family. They're cytokines that are never released from cells really, except when cells are ruptured by necrosis, and they're very powerful initiators of immunity. So they function very similar to PAMs, and they're very good at driving immunity in situations where cells die by necrosis. So I've said that now a few times. Necrosis is inflammatory, apoptosis is generally non-inflammatory. And the reason apoptosis is non-inflammatory is because there are molecules that are presented on the surface of apoptotic cells. When cells die by apoptosis, essentially what happens is there's a set of enzymes that are activated, and they result in the externalization of phosphatidylserine, as well as probably one or two other molecules that enable macrophages to discriminate apoptotic cells from healthy cells. And typically then macrophages engulf and eat apoptotic cells before they have an opportunity to release DAMs. And that's really a very key feature of apoptosis, is that early recognition and removal of apoptotic cells from tissues before they can rupture and burst open and release DAMs. I'm just going to show you some morphologies now, what necrosis looks like, a couple of little movies, what apoptosis looks like, and I've already said. So somebody's asking a question there. Yeah, you want to just shout out the question?
Okay, all right. Just a quick question before you move on. So now you talked about the various form of cell death with those that are inflammatory and non-inflammatory, and knowing fully well that this inflammation and inflammation cell death has different, some release dams, some doesn't release dams. So the question I have is, what now majorly determines how a cell dies? Is it the release of dams or PAMs or the receptors or what majorly? What determines the type of cell death that happens majorly?
Sorry, what is the type of cell? Can you repeat the last bit of the question?
Okay, when the cell becomes transformed or infected,
Yeah.
and then, so what determines the type of cell death that this particular cell is going to undergo? Maybe necroptosis or pyroptosis or apoptosis. Is it the release of dams or any other thing?
No, I mean the mode of cell death that cells undergo really depends on the stimulus the cell is exposed to, and you know, the precise kind of circumstances that the cell finds itself in. That's what determines the mode of cell death. So typically, if cells, for example, are engaged by a PAMP, and then it will upregulate inflammasome constituents, and that may really result in pyroptosis if the cell then receives, you know, a membrane damage. To be honest, it's a complex question in terms of what determines whether cells die by apoptosis or pyroptosis or necroptosis. I don't believe really there's good evidence to suggest that happens in any physiological circumstance. Typically, we only see that when cell death is in the cell death machine is inhibited by caspase inhibitors. But again, it's a bit of a long-winded debate. So fundamentally, I think the major choices for a cell is die by apoptosis or conventional necrosis, or pyroptosis. So what dictates the decision between necrosis, conventional necrosis, and apoptosis is really the intensity of the stimulus. If the damage is very extreme, it's conventional necrosis. If the damage is less severe, it's apoptosis that really we see in most cases. And then pyroptosis is the other alternative outcome, and that really depends whether there is a detection of a PAMP, which upregulates the inflammasome, you know, that detects that PAMP, along usually with some kind of damage or injury to the cell membrane, usually as a result of the infection, you know, that perturbs homeostasis. And that's a particular infection scenario. So to get pyroptosis, you need an infection, basically an active infection. But infection aside, it's typically the concentration of the dose of the stimulus that the cell is exposed to. So if it's a cytotoxic drug, high doses of drug cause necrosis, low doses cause apoptosis. If it's temperature, again, high temperatures cause necrosis, or intermediate apoptosis. And that's true of most stimuli, gamma radiation, you know, UV radiation. You typically get apoptosis at high intensity doses. So, necrosis at high intensity, apoptosis at more moderate doses. So I hope that answers the question, but we can return to it at the end. So I'll
Yes. Yeah, okay. Thank you.
There's another question, Natalia.
Yes. So I just want, because I was thinking about what you said about death programming. So I was wondering if we cannot, let's just see, if we cannot control cell death, what is the risk for the organism? Because you were talking about programming cell death, and then I was thinking about if we cannot control this, what happened for the organism?
Well, almost certainly the organism will not survive. I mean, I showed you earlier when, for example, FAS receptor was mutated, just one single means of initiating programmed cell death. And typically what happens is those mice develop an autoinflammatory condition by about 16 weeks. of age. And they have a
Mm.
massive accumulation of extra T cells and B cells,
Okay.
because every time you initiate even a mild immune response, you get an expansion of T and B lymphocytes in response to the antigen. And then those extra T and B lymphocytes, normally they die by programmed cell death. You don't need all those extra cells. You just need them while you're fighting the infection.
Okay.
And they die. And they die by programmed cell death. That's dependent on the FAST receptor to turn on that programmed cell death. So I showed you the size of the lymph nodes
Mhm.
in a mouse. They increase 50 times their normal size by 4 months of age if they can't do that.
Mhm.
But that's one obvious consequence. You know, very quickly, you're going to
Okay.
develop cancers all over the place in the body, because, you know, cells suffer DNA damage every single day in surface exposed areas of our body. You get, it's been estimated, you know, well, these estimates vary, but certainly dozens of DNA damaging ionizing events every day in many cells.
Mhm.
And if those cells were not eliminated, we would get cancer from a very early age in
Yeah.
our bodies. And also in other situations, we would have constant inflammation in our tissues. So, you know, when you have a flu or a cold, you know how good you feel because you're fighting infection. Well, imagine that amplified, you know, 100 times.
Okay.
That's how good it would be
Mm.
if you had no means to control cell death. We would
Okay.
have constantly inflamed tissues. We wouldn't be able to eliminate damaged cells. We wouldn't be able to eliminate cells, you know, that are only transiently needed in the life cycle. And also we wouldn't be able to sculpt tissues during development. So you couldn't have multicellularity regulated cell death, really.
Okay. Mhm.
So, I mean, what's
It
just
would
as
be
valid a question is, imagine we couldn't regulate cell division. What do you think the outcome of that would be?
horrible.
Yeah, pretty horrible, I think.
Yeah.
So the same applies to regulating cell death. You know, they really go hand in hand to counterbalance one another.
Okay.
So, okay.
Mhm.
All right. So to just look at a bit of the morphology, necrosis, what's it look like? Well, here, as I mentioned already, necrosis triggered by things that are severe. So high doses, high concentration, extremes. And this is what it looks like. Typically cells, they get rapid membrane damage. They start to swell. They eventually, you know, burst and release their contents. And normally that happens very swiftly within an hour of receiving the, you know, the signal or being exposed to that. And this is an example. These are cells, you know, in tissue culture exposed to high concentrations of hydrogen peroxide. And all the cells die. This is shot over a course of an hour. All cells die really at the same time. They just swell and burst and release their contents. You can, I hope, see, you know, blebs occurring, and then the membrane, you know, just swells and ruptures. And at the end, all we'll see is just nuclei sitting there, kind of this empty corpse of a cell. All of the cytoplasmic contents have been released. And those cells are all 100% dead within an hour. And, you know, that is very characteristic of things that trigger necrosis. It's very sudden, very swift. All the cells are affected. Oops. Okay. With apoptosis, it's normally more physiological triggers, lower concentrations. You can, in fact, get cells to die by apoptosis by just withdrawing growth factor. They're not injured. They just don't have growth factor. You can add TNF. TNF doesn't injure the cells. It just engages cell surface receptors. FAST doesn't injure the cells. It just engages cell surface receptors. So things that induce apoptosis are typically much more mild. The cell senses that stimulus, and then it turns on its own cell death machinery, which we'll see. Before I show you what apoptosis looks like, just a little bit of the history. The term comes from a paper published by John Kerr, Andrew Wiley, Alistair Curry in 1972 in the British Journal of Cytology. There were a group of pathologists that realized that cells died in lots of situations in a manner that didn't look like necrosis at all. They recognized a particular morphology, which is hypercondensed DNA, fragmented nucleus, and kind of blebs on the surface of the cell. And without having any evidence, they suspected very strongly cells with this morphology must be dying in some kind of programmed manner. And they realized that because of the circumstance the cells were dying in. So they proposed the term apoptosis, derived from the Greek, meaning to drop or fall away. And they envisioned it as very similar to the way trees lose their leaves in autumn. The leaves fall off. It's highly regulated. It doesn't mean the tree is dying. It's just a process of renewal. In their case, they were thinking of apoptotic cells help to renew the tissue. And it was a natural part of the life cycle of tissues. This is what apoptotic cells look like. So again, top row here is healthy cells, and then the next row here are apoptotic cells. You can see that the nucleus becomes very condensed and it's fragmented into small pieces. And that's typically what apoptotic nuclei look like. It's very characteristic. On the bottom are necrotic nuclei. They don't show that hypercondensed morphology. They certainly don't show that fragmentation of the nucleus that you see during apoptosis. And that was really the first characteristic of apoptotic cells that was recognized. And on the right-hand side here, you can see what you see if you isolate nuclei from apoptotic cells, you find this ladder-like pattern of chromatin, because what's happening here in apoptosis is that the DNA has been hydrolyzed at internucleosomal sites. So you get this complete hydrolysis of DNA within the nucleus of apoptotic cells, which causes it to condense and collapse and become also fragmented into several pieces. And that laddering of DNA is very characteristic of a nuclease that cleaves between histones. So it leaves 200 base pair multiples, and that's a characteristic of apoptotic cells. Apoptotic cells also undergo this membrane blebbing. Those blebs also bud off into apoptotic bodies. These are still all intact. There's no leakage of the cell. There's no release of contents. And at this early stage, macrophages actually are able to recognize changes that take place on the apoptotic cell and eat the apoptotic cell. And so there, again, in the center is an apoptotic cell. Macrophages can recognize those plasma membrane changes and come and remove that cell. Remember, I told you earlier, a fundamental rule of multicellular organisms is you don't eat your neighbor. Well, in apoptosis, cells give permission to be eaten by their neighbors. And again, that's very unusual. Normally, cells don't eat each other within our bodies, but during apoptosis, an exception is made for that. That's what the process looks like. These are cells, again, taken in my lab. And this movie is, the last movie I showed you was shot over an hour. This is over 24 hours. And the cells are independently embarking on the process of apoptosis. And you can see it's very active. Cells retract, they start blebbing, they're still alive. At this stage, they would be recognized by macrophages as being apoptotic, and they would be eaten if macrophages were there. And of course, some of them just refuse to die. They're just sitting there while their neighbors are dying, and they're not dying. And, the molecules that dictate whether cells, live or die in response to the same concentration of drug, of course, are very interesting from the point of view of cancer therapy. And we know what those molecules are now. The BCL2 family helps for cells to basically make that decision of whether to die or not to die in response to a particular challenge. So if apoptotic cells are left in culture, for a protracted period of time, eventually they undergo what's called a secondary necrosis. But that's not typically visualized or seen in vivo. That's a kind of an in vitro artifact situation where eventually, of course, they will, lose control over their, membrane permeability and subsequently rupture. And that cell is finally embarking on the process many, many hours after all of its neighbors have individually initiated apoptosis. So apoptosis looks very different from necrosis, and typically cells initiate the process independently. Here we've induced all of the cells in the dish to undergo apoptosis by exposing them continuously for 24 hours to a high concentration of drug. But of course, in tissues in vivo, Apoptosis is very difficult to find because cells undergo the process individually, at their own time in response to particular stimuli. Whereas necrosis in vivo is typically seen in large areas of the tissue. Things that induce necrosis typically kill thousands of cells, if not millions of cells. Andrew has raised their hand.
I have a quick question. Does secondary necrosis doesn't happen in vivo because of a first-dose process?
It probably doesn't happen very often in vivo, secondary necrosis. It probably happens only where maybe the capacity for macrophages and other cells to eat the apoptotic cells has been overwhelmed. So I don't think it happens a lot in vivo. I think it's largely an in vitro phenomenon, really. So,
Thank you.
So it's fairly well known now that apoptotic cells release signals that allow macrophages to migrate towards them, and then also they externalize phosphatidylserine, and that acts as an eat me signal. And so when macrophages find phosphatidylserine on the surface of apoptotic cells, that gives permission or licenses macrophages to eat apoptotic cells. And we can detect that using a protein which binds phosphatidylserine at high affinity called an XM5. So it's a convenient way of measuring apoptosis. So here on the bottom, on the x-axis, we're using an XM5 FITC to detect that phosphatidylserine externalization during apoptosis. So these are healthy control cells on the left-hand side. They don't bind an XM5. It's propydiamide uptake on the y-axis. That indicates membrane leakage. So if cells become PI positive here, they're necrotic. If they go into an XM5 positive quadrant, they're apoptotic. And you can see here, within 6 hours of treating cells with an apoptotic stimulus, a lot of the cells have started to bind an XM5. They have phosphatidylserine on the surface, but they're not leaky yet. We can see here at 12 hours, virtually all the cells are binding an XM5, and only a minority are going into the leaky necrosis quadrant. Eventually, after 24 hours, all of the cells will be in this double positive quadrant. But an XM5 is a convenient way to label apoptotic cells as a result of that phosphatidylserine exposure. So again, to summarize what I've said there is when a cell dies by apoptosis, it's effectively non-inflammatory because it releases find me signals, which allow macrophages to migrate towards them. And also it displays eat me signals that allows the macrophage to engulf the apoptotic cell and dispose of it before you have DAMPs being released. Whereas in necrosis, cells rupture very suddenly, they release DAMPs, and DAMPs are very powerful immune activators, and you get inflammation. And that pretty much in a nutshell is the major difference between apoptosis and necrosis. Apoptosis is non-inflammatory or even anti-inflammatory, and necrosis is pro-inflammatory. And that's why you need to regulate programmed cell death to minimize necrosis, which of course you want in a situation where it would be appropriate to have an immune response. So pyroptosis, where you've detected a pathogen, you want an immune response. And so there's a type of programmed necrosis called pyroptosis, where you deliberately release DAMPs as a means of ramping up an effective inflammatory response. And cell execution, involuntary apoptosis, where a cell maybe has its cell death machinery paralyzed by an infectious agent, and so it needs help to turn on its programmed cell death machinery. And in this case, you have an NK cell or a CTL that basically introduces molecules such as granzyme B, granzyme A into the target cell to turn on the target cell programmed cell death machinery. There's CTL killing. So all the flattened cells here are targets, and you're going to see this is a CTL, an NK cell actually coming in and basically inspecting various cells. And eventually, I think it's going to kill this cell by degranulating onto the surface of the cell, and which is doing now. And then that cell you're going to see dies by apoptosis shortly. There we go. So that's a kind of a very surgical way of eliminating an infected cell or a cell that's displaying non-self peptides. Now, I don't have time to go through the specific cell death machinery, but we know very well exactly the molecules that regulate cell death. There's lots of reviews out there, including by my own lab, on how the cell death machinery is wired. I'm just going to tell you that, you know, some of the work came from genetic studies in C. elegans, you know, some of the work that helped to unravel how programmed cell death was regulated. And then the genetic framework was really discovered in C. elegans by doing screens for genes that regulate cell death in that nematode worm. And in the nematode, it's convenient to look at events because there's 1,090 cells that are born during development of the worm. And every single one of those cells, you know, was tracked by John Sulston and Bob Porwitz and Sidney Brenner's lab, so that a cell fate map was generated. And they realized that during development of the worm, 131 cells always died. They always died at the same time, same places. So clearly, cell death was obviously being regulated in this situation. And then the genetic screens in the worm, and they found that there were worms that they could identify that had too much cell death or too little cell death and so on. And they called these cell death effective genes, SED genes. And when they boiled it all down, they found that there were two critical SED genes, SED3, SED4, that promoted all cell deaths in the worm. And then a major gene here, SED9, that could block cell death. And then it turns out that that framework has been broadly conserved through development, you know, in flies, in mammals, and so on. And essentially what happens is SED4 is a platform for activation of this molecule here, SED3, which is a protease. And in SED3 is a caspase. So in mammals, these caspases are conserved, there's just more of them. So fundamentally, what happens in programmed cell death is you want to get proteases activated. And so to switch on the cell death machinery, we have different activation platforms to switch on proteases. And these proteases are called caspases. And in the worm, this SED9 molecule, which is basically the homologue, the worm homologue of the BCL2 family in mammals, the SED9 sequesters the caspase activation platform away from the caspase. And to get cell death in the worm, you must displace this SED9 BCL2 family member away from that. And so then SED4 can bind SED3 and activate it. And fundamentally, that's what happens in mammals. And Bob Porwitz and John Sulston and Sidney Brenner received the Nobel Prize in 2002 for their work on the genetics of programmed cell death in the worm. In the fly, it's quite similar. You have more caspases, more SED3-like molecules, just still one SED4 homologue that's involved in activating one particular one, drunk. And then there's some differences in the fly I don't have time to go into. And in mammals, it's broadly the same. Again, with some elaborations, you have caspases, i.e. SED3 homologues. You have a SED4 homologue, which is called APAF1. And then a SED9 family, which is the BCL2 family. And then an EGL1 related family called the BH3 only family. So there's lots of additional members, but effectively, again, what you're trying to do is activate these proteases called caspases. And there's three major routes to doing that in mammals. And again, you know, you can read about that in detail, but the three major routes to activating caspases are through either death receptors, that's called the intrinsic pathway, or through the intrinsic pathway where various stress or damage signals are detected by BH3 only proteins. They permeabilize mitochondria, that releases cytochrome C, and then you get this SED4, SED9 complex activated. It's called APAF1 in mammals. And it's called the apoptosome, and then that activates multiple caspases downstream. So you can see here what you're doing is you're activating caspases using either surface death receptors or sentinels of damage or stress internally. And then also CTLNK cells, they deliver enzymes in that can activate either caspase 8 or BID to to directly engage with cell death machinery. So they're the three major pathways to activation of programmed cell death in mammals. And as I said, there's lots of reviews you can read about that. And then once caspases get activated, they basically coordinate the events that take place in programmed cell death to result in externalization of membrane signals on the surface. And again, in the interest of time, I'm not going to go through all the details here, but you can read that in plenty of reviews that have been published. There are thousands of proteins that are cleaved by caspases during programmed cell death. Not all of them are necessarily particularly relevant, but some of them are. And we've linked some of the caspase substrates to exactly what happens during apoptosis. I'm going to skip that. And I think I'm really just going to, well, I'll kind of finish on this. I think a major decision point in cell death in mammals is this interplay between the pro-survival BCL2 family and the pro-apoptotic BH3 only proteins. They sense various types of stress or damage. And if sufficient numbers of BH3 only proteins are activated, they permeabilize mitochondria. And that's a really, key event in the decision to die. And the pro-survival BCL2 family, they stop that mitochondrial permeabilization from happening. And we know that's really important for example, if we want to improve chemotherapy, we've got to basically find ways of increasing the likelihood of back channel opening, which results in the downstream activation of caspases. And again, I'll skip that. And I think just finish with that, that we believe that caspases are more concerned with controlling whether cell death is inflammatory or non-inflammatory rather than controlling whether cells die or not. They're really regulators of inflammation, not really regulators of cell death. We've written about some of this in reviews over the years in Molecular Cell, and hopefully we'll have a review in Molecular Cell as well on similar theme scheduled for later this year, where we discuss some of those issues. So ultimately what happens is in programmed cell death is you're activating caspases in order to control the outcome in an inflammatory sense. You want to either produce non-inflammatory cell killing where cells are removed silently, or in some cases you want that cell killing to be inflammatory where we trigger a powerful immune response. And that's one of the major reasons for regulating cell death, is really to regulate its inflammatory consequences. And I mentioned at the outset, if you get an imbalance, too little cell death, there's lots of diseases that result. So it's really important to be able to regulate cell death in lots of different circumstances. There's still a lot to do. The cell death field is quite a mature field now, but there's still lots of things really we don't understand, not so much in terms of how cells die, but when they don't die, what are the consequences of that? When cells have been, for example, stressed or damaged and they've recovered, does that matter? Do they just go about their normal business now and resume life, or does that stress or damage, instigate processes that could actually be quite detrimental in the context of a tumor, for example? That's a view of space from the Hubble telescope. That's not a view of space from the Hubble telescope, but I think it's quite similar. It's actually just one small portion of the cell, just visualizing actually about 3 different proteins. And when you consider there's about 12 to 15,000 proteins in the average cell, 15,000 different proteins, and probably 100,000 copies of each protein, cells are immensely complex, and there's a lot still to learn. And I'll just finish by saying, I think any of you who've had experience of this already probably know this, but research is amazing. It's really something that it's a privilege to be able to do. It's a very challenging job. It can be very frustrating, but it's incredibly stimulating and motivating and fascinating. It's a lot better than working in a coffee shop, as I often say. Not that I'm running down working in a coffee shop, but that seems like hard work to me. Whereas doing research is much more fun. Okay, thank you.
Thank you so much, Shay. I'm always get amazed and more motivated with your talks. You have a particular way to touch our scientific hearts and mind. So I hope all the students also get the same feeling I do all the time. Andrew, you have a question?
Is evolutionary speaking, is not more advantage to the cell invest in the repair mechanism instead of a dead cell mechanism? Which one is more expend more energy to the cell?
I think it's a lot easier to kill cells than repair them. That, there's so many ways that cells can get damaged. And when you think about it, I just said that I finished up by saying that cells are immensely complex. You have literally millions of proteins in a cell. You have this huge genome, multiple organelles. It's a very delicate, much more complex than a very high precision Swiss watch in many ways. And the only advantages you have in the body is that you have billions and billions of cells, trillions of cells indeed. So it's relatively easy to build another cell because we have like a process for doing that, rather than repairing, many different types of damage that could happen to a cell. Because the problem is, when you damage something so complex, the potential threat to the whole organism, if that repair is faulty, it can be catastrophic. And we know cancer, for example, is very common. Probably about 50% of it will experience either cancer ourselves during our lives, as we, the human populations live for longer, or certainly we will definitely know somebody who has cancer during our lives. So, that's the problem. If you try to instigate a repair, and that repair isn't completely accurate, then the chances are, a cancer will develop, and that cancer could well threaten, obviously, the life of the whole organism. So I have to say, thinking about it, it seems to me a lot easier to just simply say, well, will I engage in a lengthy repair process trying to make that repair completely 100%, or will I just get rid of that cell and I'll just make another one, because tissues are dividing anyway. It seems to me a simpler process to allow the normal process of cell division, which clearly cells have perfected, during evolution, whereas repair is so dependent on the precise nature of the injury and how many different injuries have the cell got and so on. It's, I think it seems to me that it's easier to simply kill that cell and replace it with a fresh one. So that would be the way I would view it. But of course, repair does happen to some degree, but typically you see that repair is more relied on in post-mitotic tissues, in tissues that don't receive very much injury, where the cells basically are not dividing very frequently, or the job of that cell is a very complex one, like a neuron. A neuron's role is bound up in making connections, and then when it's made those connections, it's quite valuable. If neurons have innervated with lots of other neurons, you can't just get rid of that cell and then a new cell will immediately make those connections again. Those connections have been learned. So typically we see in post-mitotic tissues that don't divide any longer, that there is more likely repair will be engaged in those tissues. But for less complex tissues where there's lots of cell division going on in the tissue anyway, I think it's easier simply to kill that cell and replace it with a fresh cell.
Thank you, Professor.
Marceli?
Hi, thank you, Professor, for your presentation. I have one question. And the signal of HME exposure phosphatidylserine can turn off or always is turn on? And how long does it take for the cell in apoptosis actually die? When you see phosphatidylserine exposure but don't see any difference in cell viability. I have this problem in my experiments.
Yeah, when do you first see phosphatidylserine exposure? Typically within a couple of hours of applying the stimulus, but it does depend on the stimulus and, you know, the intensity of the stimulus. And then, you know, you're saying, when does an apoptotic cell lose viability? Were you asking that?
I understood that she asked whether the sterilization of phosphatidylserine could be reversible and then the cell don't die. Is there any situation?
There have been one or two claims that it's reversible. I have to say I'm not really convinced by the evidence that's been proposed in those papers. But generally speaking, I would say in the vast majority of cases, it's not reversible. When a cell gets to the point of externalizing phosphatidylserine, it's really committed to the process. We know this is caspase dependent. You know, so there has to be sufficient caspases activated at that stage. And when you've got a kind of a certain concentration threshold of caspase activation breached, typically the process is fairly irreversible. At kind of very low levels of stress or damage, you can find certain fraction of cells that maybe exist in a more gray area where they're right at the threshold of committing to cell death. And in special circumstances, you may be able to allow a cell to survive that wouldn't otherwise have survived. And that's usually by intervening in some way, like blocking caspases. So for example, you can expose cells, for example, to low doses of cisplatin, that's a chemotherapeutic drug, or radiation, or some other damaging stimulus. And you can find conditions where maybe 50% of cells die. And then by blocking caspase activity, you can find that, you know, only 40% of cells die. So there's a 10% there that have now survived that wouldn't have survived if you had allowed caspase activation to happen. But in those circumstances, what we find is what happens to the cells is they've clearly been injured sufficient to start to activate the cell death machinery. We've allowed them to survive by blocking caspases, but they've been clearly wounded. And normally those cells become senescent. They never resume a normal, you know, healthy lifestyle. They become post-mitotic. They have features of what's called senescence, premature replicative senescence. And I kind of see them as almost, you know, mummified. They're not dead, but they're not really properly alive. And that process of senescence is actually is quite fascinating. And we're looking at it quite a bit in the lab at the moment. And as indeed, of course, there are a lot of other labs. But we know that if you rescue a cell on the threshold of cell death, it doesn't just become viable again. It actually goes into senescence, which is like a state of paralysis, where it's damaged and it's not really properly recovered, but it's not dead. Yeah, I hope that answers your questions, but basically, no, most of the events are not irreversible. You know, when the cell commits to a threshold, and then it usually completes the process.
No, thank you for answering my question.
Karina.
Achei, I really like the way you deeply think about the questions and put this in a very simple way. So, but for me, it's quite easy to understand the role of the intensity of stimuli to induce or necrosis or apoptosis. But when you think about necrosis versus paroptosis, it's quite not simple, because we know that not only during infections, but also during sterile conditions, such as particulated material or some some damage of organelles can induce this kind of necrotic cell death, regulated necrotic cell death, such as paroptosis, and how you can separate this accidental necrosis versus paroptosis. I mean, I don't believe in this accidental necrosis, because I think almost all these stimuli can induce paroptosis, and paroptosis is also so fast as accidental necrosis.
Well, if you literally take a lighter and put it to your finger, you're going to get necrosis. Okay, that's not paroptosis. If you get a hammer and smash your hand with it, that's not paroptosis. Okay, that's conventional necrosis. So I guess paroptosis, if you want to boil it down, is a caspase 1 dependent rupture of the plasma membrane via gasdermans. That typically only happens where you get an inflammasome activated, and you typically only get an inflammasome activated where you detect the pathogen.
No, you can induce inflammasome activation in response to membrane damage.
No, but
Plasma efflux can induce inflammasome activation.
But you need priming with LPS or another PAMP.
It depends on the cell. Some cells can have this expression, the physiological expression of caspase 1 and NLRP3 and macrophages, for example. So not all cells can die by paroptosis, but some of them can die.
But again,
I see this stimuli.
I see the majority of those cases as a two-signal situation where you have the priming signal 1, which says, okay, this is evidence of infection, but I don't see danger yet. I don't see injury yet. So you prime the system with a PAMP in the majority of cases, and now you prime those cells by upregulating, NLRP3 or other NLRs. And now if you receive any kind of membrane damage or homeostatic perturbation, now that's signal 2, and that can be any kind of thing that perturbs membrane damage. So I see it really as just a modified form of a necrosis, only in this case, you've primed the system by upregulating pathogen sensing molecules, pathogen sensing platforms, and that now when you get a perturbation, you now drive this caspase 1 dependent, release of the damps that you've upregulated. So the priming step also upregulates IL-1 beta, IL-36, and many other IL-1 family cytokines.
You're right. You're right. Yeah, you're right. For the majority of cases, conditions, but now you see some of these inflammasomes are expressed in many cells, not only immune cells. And in some cases, if you have a potent stimuli, you can induce inflammasome activation and paroptosis, even without this TLRs, priming, and so on. So it's not quite simple.
I think that's the exception. I mean, in trying to understand biology globally, take a step back and say, well, look, what works in most cases? And in those cells you say where you don't need priming, I would suspect as well it's not very inflammatory, because those cells, either they already have high levels of IL-1 family members, but most cells don't, Normal priming step dramatically upregulates alpha beta. I mean,
That's right. That's right.
so if the cell hasn't been primed, then there's very little alpha beta to release. So,
That's right.
or indeed IL-18 and so on. So I think in most cases, pyroptosis is just a modified type of necrosis that says, okay, I've detected a pathogen. So if the cell is damaged, I'm going to release all of this DAMP, all of this cytokine. And in most cases, when you detect LPS, the cytokine that you release is IL-1 beta. And so I see this as a way of driving a very aggressive inflammatory response to pathogen infection. That's really what pyroptosis is, by using necrosis as a means to dump out that DAMP. Once you detect signs that there's a bacterial toxin around, or the bacterium is perturbing the kind of tissue physiology so much so that now your membranes are being damaged and so on. So we just detected a little bit of pathogen of PAMP without the qualification signal of danger, then it's like, okay, I've detected a little bit of bacterial infection, but the bacterium isn't causing much damage. So I'm on alert, but I'm not going to, burst open and release lots of cytokines. And that's how you, I suppose, deal with mild infections or commensal pathogens. You dial things up, but you don't go to the second stage. So that's the way I would look at it.
Yes. That's fine. Thank you, Shay.
So in a way, to use another analogy that I often think of, I think an immune system, or just like in society, if you have a little bit of trouble, you call the police. If you have a lot of trouble, you bring in the army. And pyroptosis is kind of, activating the army. Apoptosis is maybe using the police to just.
I like that. I like that.
Yeah.
It's strange, Seamus, that you mentioned that, because we have this way of illustrate the immune system or whatever biology with this association with the society, or how society operates. And it just reminded me that in the first question that Andrews asked you, it's exactly the opposite. So the organism, the nature, do not operate as our society in terms of looking at the individual and try every single way to rescue this individual, but rather operates at a large scale to maintain a functional society, as you just mentioned. So it's sometimes the illustration is the way that we work as a society, but not always, huh? Sometimes it's the opposite.
Depends on the political governance structure, I would say.
Maybe.
So,
maybe
there can be collectivist politics that says the individual doesn't matter. It's all about the collective. And then there's extreme kind of liberal, where it's like all about the individual and anything goes, and that can be,
right.
Both have their strengths and weaknesses, clearly. But
all right, Andrew, another one.
Before Andrew, asks a question, I would like to know how Lucas is represented twice on the Zoom.
Yes.
So is that your twin brother?
Twin brother that moves exactly the same.
Yeah, they're very close.
It's because my computer's mic is with a technical problem, so I had to join through my mobile. So I could, if I had a question, I could ask it through my mobile. And actually, I was looking for pyroptosis, and the meaning of pyroptosis is like fire, and pyro from the Greek fire. So there is inflammation in his chest. So probably, I think this is a sign that is associated with inflammation and infection. There is membrane rupture as well, which is not really come into apoptosis. So I think there are lots of fissures that indicate that apoptosis is quite different from apoptosis. Just like adding
I wouldn't always read too much into the word that's used to describe a process, because sometimes it can be quite superficial. Somebody introduces a new term. Sometimes it's not often that well thought out. There's been lots of program cell death modalities that have been suggested over the years, and some of these are getting to the point of ridiculousness, to be quite honest. Almost defining the cell death by the stimulus used to achieve the cell death. And so we may have, for example, cisplatin apoptosis, staurosporin apoptosis. I mean, every different drug could potentially induce its own individual cell death modality. So I happen to think there are fewer modes of cell death that have been proposed. There's all sorts of different regulated cell death modalities that have been proposed in recent years, and I think a lot of them don't really stand up to scrutiny, or there's very little evidence that they really occur in vivo. So that's an ongoing discussion, I think. So, for example, necroptosis for apoptosis, not a lot of evidence that they actually take place in physiological scenarios, but that's a debate for another day.
Yeah. It was just like because of fever, and I just related that to inflammation. So if there's inflammation, it cannot be related to apoptosis. So there was just a path through, like, separating these two cell death mechanisms, which is like how
when the term apoptosis was coined, there was absolutely no interest in inflammation as an outcome of that process. In fact, the interest in cell death and inflammation really, only started to intensify from around 2010 onwards, thereabouts. People in the cell death field weren't really interested in inflammation. They were seeing as very separate fields. Progressively over the years, the study of cell death and inflammation has become more and more intertwined, because they are very intimately linked, as I've been saying. So yeah.
Thank you.
Thank you. There was, I think, somebody else, Andrew, was...
Yeah. So in necrosis, the cell does not participate, mainly because the process is too fast to be programmed, and the stimuli is too fast.
Yeah, so except in the case of pyroptosis, which is a type of regulated necrosis, in most forms of passive necrosis, the cell doesn't participate, usually because the damage is too severe, and events proceed very, very rapidly. So if you take, I mentioned that earlier, if you take 500 millimolar hydrogen peroxide, cells will die within an hour. If you take 100 millimolars hydrogen peroxide, at 500 molar, cells will die within an hour, and there'll be necrosis. You drop the concentration down to 100 millimolar, cells will take 12 to 24 hours to die, and there'll be apoptosis. And that's what we've observed, generally speaking, with most damaging agents. Lower doses of the same agent induces apoptosis. So the cell has sufficient time to sense the damage and then make that decision whether or not to engage its cell death machinery. And higher doses of the same stimuli typically induce necrosis. So 44 degrees, you get necrosis like within an hour. 42 degrees, you get apoptosis. Alcohol 5% induces apoptosis. Alcohol 10% induces necrosis. We've done this for a lot of agents. There's a paper, Lennon et al., very, old paper actually. And in that paper, we titrated like dozens of drugs and other stimuli, and that's the general thing. As I said, the exception is if there's a pump in the system, you dial up your pyroptotic machinery. And now if you get a damaging stimulus, typically you very rapidly activate an inflammasome. And that actively bursts open the cell through caspase-1 cleavage of gasdermin D. So.
Thank you, Professor.
Ricardo?
So Seamus, could you comment a little bit?
I was just saying hello to Sean, Alexandra. Welcome.
Ah,
so we
hi guys, sorry to
Yay.
interrupt.
Hello, Sean.
We're still
Hello.
finishing the discussion with Seamus, then we will get to you. All right.
Okay, great.
you.
All right.
Stay with us.
Yeah, sure.
So Seamus, could you comment a little bit on the redundancy of the pathways there? Because, you know, we have caspase 3 and caspase 7. If you knock out caspase 3, most of backgrounds, you don't see much of an effect. We have a lot of BCL2 family members that are induced at the same time. So I would like to hear from you about what do you think of, in terms of redundancy of the pathway?
Yeah. So I guess, you know, that's common enough in important pathways that there be degree of redundancy in the system. Caspase 3 and 7 seem to be enzymatically pretty redundant in terms of their substrate specificities, but they're not necessarily expressed to the same degree. In our experience in many cell types, it's 90% caspase 3, 10% caspase 7. So I do think there's a degree of redundancy there that's, you know, built in, you know, to ensure that the system has robustness. I think we see that in quite a few biological pathways. In terms of BCL2 family, you know, again, there's, I think, of course, all of them can achieve ultimately the same goal to a greater or lesser degree, which is activation of the back-to-back channel or blocking activation. And I think that's because you know, there are many different routes to you know, damage cells or open the back-to-back channel. So all those BH3s are specialized towards, you know, sensing different types of stress or damage. And so, you know, for example, BID can respond to activation of, you know, death receptors and is the only one that can be cleaved by caspase 8. So BID can, you know, be a conduit between activation of the TNF, faster trail receptors, and then opening of the back-to-back channel. Can also be, it's the only one that's processed by granzyme B. So it can respond to CTL or NK mediated attack. BIM is one that responds to growth factor deprivation. BIM is also upregulated in the context of ER stress. And, you know, so they're all specialized or skewed towards particular types of stress or damage. So I'm not sure that's quite redundancy. I think that's more, let's say, you know, there's been an elaboration of members of the BH3 only family so that the different types of damage that cells could experience can be detected efficiently. Now, you may see multiple members of that family activate in response to a stimulus because that stimulus causes multiple types of damage. And so you're getting multiple BH3s mobilized because the cell has simultaneously received ER stress. That's also called protein misfolding. You know, that's also called reactive oxygen production. And so you have BH3 only proteins that respond to all three of those different types of damage being mobilized. And that looks like redundancy, but in fact, those, you could potentially activate just one of those BH3s if the stress was very specific. But in a way, life isn't like that, you know, because most forms of damage is never just kind of surgically precise. You know, unless you're literally irradiating a patient with, you know, a very precise radiation beam. When we get damaged or injured, typically you get multiple simultaneous forms of damage happening at once. And that's also true of cytotoxic drugs. A lot of drugs we think just do one thing. That's not true. They do multiple things. It's just we know they do one thing, or at least, and I'll give it a good example. Paclitaxel, it's a microtubule freezing agent, and it's very commonly used to treat, breast and ovarian cancer. And we think of that as a microtubule disrupting agent. But to our surprise, a couple of years ago, when we were studying this, we found it was very good at driving inflammation. And when we, disentangled how that works, we realized that Taxol is also very good at inducing ER stress. How it does that, we don't know, but it does it very well. So it's thought of as a microtubule disrupting agent, but it's also very good at initiating ER stress. And it's the ER stress pathway that causes it to promote inflammation. That's another thing it's not known. Clinicians don't know that Taxol promotes inflammation. And, they're using it to obviously to kill, as I say, breast cancer, ovarian cancer. But it has this unfortunate collateral side effect of triggering inflammation as well. That is quite undesirable in the context of cancer treatment. But there's no avoiding that. You can't have one thing without the other. So there's no perfect drug. So I hope that's answered your question, but I think some of the redundancy is, it basically makes the system less prone to, viruses or, just gene inactivation causing a complete loss of function of the system. And then some of that redundancy between BH3s or BCL2 family members is just simultaneous activation of, I think, some of those family members by different types of stresses. But I think you do need, redundancy in the system to ensure that the cell death machinery isn't, inactivated or neutralized frequently by infectious agents or just through gene mutation, because it's so fundamentally important. So,
Shay, just to complement Ricardo's, because you touch in another concept, which will be specialization. So it seems to me that the more complex the organism, more specialized molecules or cells, tissue. And in this case, it looks to me more like, at least for the BH3 only, for instance, would be more specialized pathways than redundancy. So you have a comment?
Yeah, that's what I was saying. So I think what looks like redundancy is more the activation of multiple BH3s in response to different kind of forms of stimulus that maybe it looks like you're activating just one pathway, but you're activating three pathways there. And so that the BH3s are specialized towards each of those pathways is being activated in parallel.
Yeah, but what I mean is that in your experience, these specialized, would we have like a gain in evolution or in the function to be more specialized, or it's just something that happened and not really a gain in biological terms?
Well, I think in evolution, we typically see a move from simplicity towards complexity. I think it's true to say. I think that's generally the trend. So with complexity becomes specialization, right? So where you had one molecule doing a job in the worm, you now might have 6 doing that in a more evolved organism. I think we see that, right? We see gene duplication and divergence, where now that it adds additional capability to that, gene family, that fundamentally they're doing the same thing, but it's done now in a more, specific manner, in a more regulated, highly regulated, more sophisticated manner, in higher organisms.
All right, thank you. Abology, do you have a problem with your video, Abology?
Okay, so my question, during your talk, you said that pyroptosis is caspase-1 dependent, right?
Yeah.
So that means if caspase 1 is knocked out, then there will not be pyroptosis. The mechanism will not, the cell death mechanism will not occur, right?
Yeah.
Okay, so my question is there any bypass mechanism for in pyroptosis to activate Gas-DMD? And then second question is, does this occur physiologically in human?
Does what occur physiologically, the Gas-DMD activation?
Yes, yes.
Yeah, absolutely. It's a very important pathway in humans, yeah. So absolutely does occur physiologically. Is there a bypass? Well, there's other ways. There's other ways to activate Gas-dermans. So caspase 8's been implicated and caspase 3 in other scenarios of activating Gas-dermin E. And that field at the moment is, moving quite quickly. There are other claims of Gas-dermans being activated by bacterial enzymes, by granzyme B. I'm not so sure of those claims. But there are other ways to activate Gas-dermans, yes. The caspase 1 pathway activating Gas-dermans is absolutely physiologically very important. There's no doubt about that. And I'd say that is the key pathway. And there's other caspases, caspase 4 and 5, that have been implicated as LPS sensors of intracellular LPS that can be directly activated as a result of binding to LPS. That's the non-canonical LPS sensing pathway. And, whether they can activate Gas-dermans, I'm not entirely sure, to be honest. But they certainly can activate caspase 1. So there are other ways of getting Gas-derman activation, but the caspase 1 pathway really, I think, is really the key canonical pathway. And I'm not sure there are humans, I haven't kept up with that literature actually, but I'm not sure whether there are humans with caspase 1 loss of function mutations. There are certainly humans that have various, elevated levels of caspase 1 activation. And those individuals typically have constitutive inflammatory conditions. So it's not good to be activating caspase 1, spontaneously if you shouldn't be activating it. So yeah, the caspase 1 Gas-derman pathway is, really important. And of course, IL-1 beta is the other key, and IL-18, the other key substrate there of caspase 1. So, and Gas-derman is activated to release IL-1 beta from the cell and IL-18.
Okay, thank you.
All right,
Okay.
guys.
Yes.
Shay, it was wonderful again to listen to you. It took 30 minutes more than we planned.
Yes.
It was amazing. We could still talk for another hour or two, and but I will ask you now to stop. And thank you so much, everybody. Let's hear. Thanks.
Thank you all.
Thank you, Shay.
Yeah. Hope to see you very soon in person again. Would be nice if it was every other week, at least.
Yes.
But
Okay.
yeah, so Karina and I miss you a lot already.
A lot.
It's been only a couple of weeks. So
All right, guys.
take care.
See you.
Bye-bye.
Thanks for the rest of the